Noswaith a chroeso. Good evening and welcome, everyone. It's a great pleasure to be able to introduce this evening's speaker. Dr. Cheney Drew is a Senior Research Fellow and Deputy Director for the Brain Health and Mental Wellbeing Division within the Cardiff University Centre for Trials Research. Cheney has a background in pharmacological research with particular reference to neurodegenerative diseases, having completed her PhD in Huntington's disease. Since transitioning to clinical research, Cheney has been involved in the development and delivery of trials aimed at evaluating interventions, particularly non-pharmacological and novel advanced therapies, in people with Huntington's disease, Parkinson's disease, and other neurological disorders, including tuberous sclerosis and epilepsy. Cheney is a member of the European Huntington's Disease Network Advanced Therapies Working Group and Stem Cells for Huntington's Disease Collaboration, both aimed at devising the most efficient and robust methods for evaluating novel advanced therapies for this rare disorder. Given the complex and experimental nature of advanced therapies, Cheney is particularly interested in how participants are approached, informed, and provide consent to be involved in these trials. This includes listening to the participant voice as a central tenet of trial design. So there will be opportunity for our participants to place questions in the Q&A at the end of this evening's presentation, and we'll try to get to as many of those questions as possible. So good evening and welcome again, Cheney. Many thanks for giving up your time this evening, and we very much look forward to your presentation. Over to you.
Thank you very much for that introduction. Welcome, good evening, everyone. I won't try and say it in Welsh. My Welsh diction's terrible. My children criticise me all the time. As James just said, I'm going to talk to you today about how advanced therapies can be used to treat neurodegenerative disease. Hang on, we're just going to move the slides forwards. There we go. So just to give you a quick overview of the types of things I'm going to go over with you today. So I'm going to spend a little bit of time just talking about what we actually mean by advanced therapies. What are they? What do they do? I'm also going to talk a little bit about how advanced therapy development differs from how we developed what we would call traditional medicine, so the medicines you'd normally get from your doctor. And then I'm going to go into a bit of a background overview of neurodegenerative diseases where advanced therapies may be useful. And I'm going to be concentrating particularly on Huntington's disease and a little bit on Parkinson's disease as well. And then the interesting bit, but hopefully for everyone, I'm going to walk you through how we might use advanced therapies to treat those neurodegenerative diseases and potentially other neurological disorders. And then lastly, just to sort of round off my presentation, I'm going to discuss a few of the key challenges that we have in evaluating advanced therapies in neurodegenerative disease in clinical trials. So to start off, what are advanced therapies? So generally, they fall into three main categories. We have gene therapies. So a gene therapy is where a particular gene is inserted into the body in some way. And we'll talk a little bit more about how that may be done. So it can be a small portion of a gene or a whole gene. And then we have cell therapies, which, as the name suggests, it is somatic cells that have been altered in some way. And that's a key difference between cell therapies and what we would just call a tissue transplant. So a cell therapy will have been altered in some way. So it changes its biochemical properties or its functional properties in order to treat and target disease. And then we have tissue engineering. So tissue engineering can be a combination of cells and tissue scaffolding. These will have been modified to enable repair, regeneration, or to replace tissues within the body. So people may have come across some gene therapies or cell therapies that are already out there as treatments. So many people will have heard of CAR T therapy, which is still relatively new, but it's a cell therapy that's used to treat cancer, where a person's immune cells are taken from their own body, but they're altered so that there's the cells on the surface, the receptors on the surface of that cell are able to recognize tumor cells more effectively. And that means that those immune cells can target tumor cells and specifically kill just the tumor cells. And in some cancers, CAR T is a very, very effective therapy. So advanced therapies have there's been an explosion in the development of advanced therapies over the last decade or so, as they may have really good use for diseases and conditions where what we call traditional pharmacological approaches, so any kind of drugs, have not resulted in disease-modifying therapies. And I'll talk a little bit more through that as we go on. So how is ATIMP development? And when I say ATIMP, it's a shorthand for talking about advanced therapies. So how does that differ from traditional medicines? So I'm just going to walk you through some steps of sort of the global way in which drugs are normally developed. So we start off by understanding the biological basis of disease. So what is the disease caused by? What changes in the body actually affect the symptoms that you see or that affect a person when they have a particular condition? And if we understand the biology, we can use this knowledge to identify what we call a drug target. Now, a drug target can be all sorts of different things. So it can be something which we know goes wrong in a disease or doesn't work properly. So that may be a particular protein or an enzyme that has a very specific function. So we can work to find drugs that change that function. And a target is also something that is modifiable. And that means we can change it by using a drug of some description. And normally you'll find this would be either an increase or a decrease in either activity, if it's an enzyme, or function. So one example is sort of for benzodiazepines, which are used for treating anxiety and all sorts of different things. So they work on a particular receptor in the brain, which has sites for the neurotransmitter it's responsible for. They work to control the rate of neurotransmission in the brain. So how much cells talk to each other and the messages that travel in your brain to make your brain work properly. But benzodiazepines bind to a very different site on the receptor, and they work to modulate that function of the receptor. So it works in a different way and it decreases the activity. And that's how it produces its effects. And then once you've found your drug target, so pharmaceutical companies have these compound libraries. It's thousands and thousands, if not millions, of different drug molecules. And they set up these things called high throughput assays, which is just a really quick way to, it's a sort of quite a quick and dirty experiment, really. I mean, it's very technical, very involved, but they set up an experiment so that they get a readout, so positive or negative, if that compound can bind to that drug target and does what you want it to do at that target. And so you'll get then a list of hits, things that do what you want to do. And then you can use chemistry and other techniques to modify the properties of that drug to make sure that it does what you want to do. And when we talk about optimizing the drug, you may want to change its chemical structure so it has a shorter half-life in the body. That means your body metabolizes and gets rid of it quicker. Or you may want to do something called increasing its oral bioavailability, which means that you can take the pill orally, which is the easiest way to take a medication, and that if you take that pill, that medicine still gets into your bloodstream and to the places it needs to go. So there's lots of clever chemistry that is done to make drugs more effective. But with advanced therapies, what we find is that they are usually specifically engineered. So, you know, they're not screened. You've identified a target, so a biological place where you think you can make a difference. And then you get advanced therapies engineered to engage that broad target. So already they're quite different from how we would develop normal medicines. So now I'm just going to give, as I said, a quick overview of neurodegenerative diseases where ATIMs may have some use. So the first one I'm going to talk about is Huntington's disease. So Huntington's disease is what we call an autosomal dominant inherited neurodegenerative disorder. This means that you only need one copy of the faulty gene for Huntington in order to get the disease. So every time a baby is born, they get one half of their genes from their father and one half of their genes from their mother. And usually with genetic diseases, or you'll have what is a recessive mutation. So both your parents need to carry a faulty copy of that gene in order for that child to get the disease. And that's a 1 in 4 chance. But with Huntington's disease, only one of your parents needs to have one copy of that faulty gene and pass it on to that child, which means if your parent has Huntington's disease, you have a 50% chance of also developing Huntington's disease in your lifetime. So Huntington's disease is caused by a mutation in the Huntington protein, which we abbreviate to HTT for short. So here I'm just getting my little pointer up. So here we have just a brief schematic of what a Huntington gene would look like. So this turquoise bit is the length of the gene, and these little blue areas here are what we call coding regions or exons. So when your gene is being turned into protein, these turquoise bits get chopped out, and it's these blue bits that are really important for providing the information for making the protein that you want to. So in Huntington's, in the Huntington gene, in this first blue bit here, it's called exon 1, we have something called a CAG repeat. And this is where the sequence of the DNA just repeats itself. So CAG, 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 CAG. Everybody has this repeat. It's in all of our Huntington genes. However, CAG repeats are quite unstable and they can get bigger. And so in Huntington's, what you see is if you have an elongated CAG repeat, you go on to develop the disease. And there's a really simple blood test that can be done where the CAG repeats are measured. If you have under 35, you're not going to develop Huntington's disease in your lifetime. If you have a repeat length of between about 36 and 39, then you may develop some Huntington's symptoms later on in life, much later on in life. But if you have 40 repeats or over, you will definitely develop the disease. And so symptoms of Huntington's come in at about midlife, and they slowly decline over about a period of 15 to 20 years. And Huntington's disease is a fatal disorder. It's particularly devastating because it runs through families. So it's the social and family impacts of Huntington's disease are really quite intense. Well, we move on. So the Huntington gene mutation that we've just shown you there, and the CAG repeat protein with a toxic gain of function. So because that CAG repeat is longer, when the protein is made, it doesn't fold in the right way. It means it can't do the things that it's supposed to do in your cells. We know that Huntington is vitally important for development, because if you take that gene out, it's embryonically lethal. And so you need it for embryonic development and the birth of a live baby. So it produces this toxic gain of function, which means it does lots of things in the cells that it isn't supposed to do, that are not good for the cells. And in the brain, that sort of cellular dysfunction leads to a very specific neuronal loss primarily in one particular area. So it causes the loss of what we call medium spiny GABAergic interneurons in a region called the striatum. So here we have a Huntington's disease brain that's been obtained post-mortem compared to a normal brain. And if you see here, this is the striatum here. So this little sort of slightly browner area is where you should have lots of healthy cells. So here is the same area in the normal brain. So you get a reduced loss of cells in the striatum, and you also get an enlargement of the ventricles here. So these fluid-filled spaces that are in your brain. And then as the disease progresses, you get this global atrophy, so the shrinking of the brain, which is why it looks so much smaller on this slide. And then the cell dysfunction and the cell loss that you find in Huntington's disease results in what we call a triad of symptoms. There's three core features. There's a motor symptoms. So Huntington's disease was originally called Huntington's chorea. So that describes what are called choreiform movements. So they're movements that can't be controlled. They look pretty random to the untrained eye. And causes problems in gait, falling over, ability to walk properly. They also cause cognitive problems, so problems with memory, thinking, and particularly in things that we call executive dysfunctions. That's like planning and being able to do sort of things like managing money and sort of household functions. So that executive function is really, really affected. And they also have behavioural symptoms as well. So there's a lot of depression, a lot of apathy, impulsivity, and sort of related behaviours. So it's really quite a lot for people and their families to deal with. And so what I wanted just to highlight, so you'll see that this is just a brief schematic of how networks in the brain function and where all the different brain regions interplay. So here we have the striatum. This is the place where we get the cell loss. But you'll see it has lots of connections to other places of the brain. So the cortex is super important for sort of your higher order functions, your motor system for controlling your movement, and the thalamus is sort of like a relay in the brain where all the information goes through and goes out again. So you can see, but the striatum's kind of in the middle here. So by losing neurons in the striatum, it affects lots of different brain systems, and that's why you get this sort of triad of effects, and it's not limited to one or two symptoms. And one of the big problems with Huntington's disease is there are few treatments available. So you can treat some of the symptoms, you can treat depression, you can treat anxiety, you can't really treat the movement disorder. Some of the behavioural problems can be treated with antipsychotics, but there are no treatments available that will modify the disease in any way. So it is still a fatal neurodegenerative disorder. And then moving on to Parkinson's disease briefly. So Parkinson's disease, probably more people have heard of. It's the second most common neurodegenerative disorder. So it affects about 10 million people worldwide. and around 135,000 people in the UK. But it has a number of different causes. So unlike Huntington's disease, where there's a single gene, we know what the cause is. Parkinson's disease is a lot more varied. So you can get genetic causes. There are genes or gene mutations that confer a risk for increased risk for developing Parkinson's disease, but there's no one gene where you can say, if you have this gene, you will definitely develop Parkinson's disease. We know pesticides have been shown to increase risk of developing Parkinson's disease. It's more prevalent in men than in women. And also there's been some research that has shown that things like traumatic brain injuries also increase your risk of developing something like Parkinson's disease. So Parkinson's is most commonly associated with problems with movement. So again, it's another movement disorder. People be very familiar with the tremor that people get, the one-sided tremor, problems with walking gait. People with Parkinson's often have freezing of gait problems, where they're unable to initiate movement in a normal way. And this is all caused by a loss of dopamine producing neurons in a region of the brain called the substantia nigra. And it's that loss of dopamine at substantia nigra that leads to that movement disorder. And here we go. This is just another schematic that I don't want you to really concentrate on, but it's again showing here that damage of the substantia nigra down here causes a lack of dopamine into what we call the caudate and putamen. So that is a structures in what we call the basal ganglia, which includes the striatum. So that's where it has a similarity with Huntington's disease. And that's what leads to the problems with movement. But it also, with Parkinson's disease, you get other non-motor symptoms. So people have problems with sleep, that you can get problems with cognition. Again, depression is another problem. So it's these diseases are really not limited to the motor disorder at all. And in Parkinson's disease, you can treat the symptoms with drugs. So dopamine agonists, things that increase dopamine transmission and the amount of dopamine in the brain. They control the symptoms quite well, but it's a gradual decline, so they only control them for so long. And there are other interventions as well available as well, things like deep brain stimulation, which involves an electrode put into the deep structures of the brain, substantia nigra, and it has a little electrical pulse that stimulates the production of dopamine, which helps combat some of the motor symptoms. So now on to the interesting bit, and how can we use ATIMPs to treat these neurodegenerative diseases? So using Huntington's disease as an example to start with, so we can use gene therapies to lower the levels of mutant Huntington, and I'll just walk you through how we can do this. So when you're in your cells, we all have a nucleus with our double-stranded DNA that you'll be familiar with. And your DNA is used to produce proteins and enzymes that allow your cell to do the jobs that it needs to do and function properly. So in order to get from your DNA to your protein, what happens is this double-stranded DNA unfolds into a single strand, and it is matched. So we can see we've got matching of these bases. It is matched by other molecules floating around in the cytoplasm of the nucleus to produce a single-stranded molecule of what we call messenger RNA. So this is sort of similar to a single-stranded DNA, but it still contains the correct code for making the protein. And then what happens is this strand of mRNA bubbles along to what we call a ribosome, and that's part of the cell sort of engineering structures, and the mRNA binds to the ribosome, and the ribosome reads that code on the mRNA, and it brings in the building blocks of the protein. So each protein building block matches a specific code on the DNA. So the CAG, that codes for one protein building block. And there are lots of different codes for about 24 different building blocks for proteins. And then you get your lovely folded protein that you're supposed to. So this part of the process, so the unfolding, creating the messenger RNA, is called transcription. And then taking that mRNA and making protein is a process called translation. So what we can do is we can build a short section of sort of single-stranded mRNA that complements or is complements the mRNA sequence, and it will bind to the mRNA molecule. And because it becomes then, this little section becomes double-stranded, is it stops this process of translation. So the protein isn't made. And that this short-stranded, this short little sequence of mRNA is known as a very tricky name. It's called an antisense oligonucleotide. And that can be used to lower the levels of a protein in the body. So antisense oligonucleotide therapies are in development for HD. So one way of getting them into the body is that they are administered via intrathecal injection. This means that they're injected into the spine in a sort of similar to how you would have a lumbar puncture. And the antisense oligonucleotide goes into the cerebrospinal fluid, which is the fluid that coats your spinal cord, and that can travel all the way up to your brain. And you remember when I showed you the picture of the Huntington's disease brain, it had those ventricles. So those are full of CSF. And so that means the ASO can get into the brain. You can't just inject these straight into the bloodstream, and you can't take them as a pill because they will get broken down very, very quickly. So to get them into the brain, this is how we do it. But it does require repeated administration. Now, it's been an effective therapy for other disorders. So for spinal muscular atrophy, which is a very unpleasant neurodegenerative condition, which is often very common in small children. It's not a common disease, but it's more commonly affects small children and can be lethal. So when that was trialed about a decade ago, it was trialed in a double-blind placebo-controlled trial. So half of the children in the trial got a placebo, so a dummy medicine. And what happens in a trial is that an independent board periodically looks at the data, mostly to make sure that new treatment you're testing is safe and it's not harming anyone. So when they looked at the data, it was so clear that the treated children were doing so much better that actually the trial got stopped because it was no longer ethical to give half of those children a placebo anymore, because it was so evident that that therapy was working. And that therapy is now available for people with SMA. So they still require repeated administration. So they have a series of 4 loading doses over a short space of time. And then they have a dose every I think it's 3 months or so. As I say, these ASO therapies are currently in trials for HD. So there are, with ASOs, they generally, for many of the trials that are there at the moment, they decrease both the levels of mutant Huntington and the levels of what we call wild type Huntington. So remember, we've got 2 copies of the gene, but only one is faulty. So you get some good protein and some bad protein. And these ASOs knock down both of those, which is why you're still seeing very early phase studies for these. But it is possible to create what we call an allele-specific ASO. So it specifically targets just that faulty copy. And so that is also in trial. Now, another way or an alternative for getting this kind of therapy into the brain is not via intrathecal injection, but it's to use what we call a viral vector or viral mediated gene therapy. So this is a really useful diagram that I've taken from the US National Library of Medicine. So here we have a virus, and so you can use different types of viruses, adenoviruses or lentiviruses. I'm not an immunologist, so I'm not going into the difference of why one may be better or not. But you take this virus and you modify it in a way that it can no longer cause disease or illness in the person. And you modify the virus's DNA to include your new gene, the one that you want to get into someone. And so you inject these adenoviruses into a person, they go into the cells, and then the virus gets to the nucleus, it offloads that new gene into the nucleus, and then the machinery of the cell in the nucleus creates the protein that you want to. So gene therapy for PD, sorry, I skipped ahead there slightly, is also possible. And there are several different ways that we can do this. So one, we can use genes to effectively replace the dopamine that is lost by losing those dopamine cells in substantia nigra. So you can use genes that stimulate dopamine production in other cells that are already in the brain region. And so this is using targeting sort of that dopamine synthesis pathway that you have in the brain, and getting the brain to make its own dopamine. You can also modify the motor circuit. So remember, I showed you there's really intricate circuits with sort of negative and positive feedback loops. So you can exploit that and selectively stimulate other areas of that circuit to compensate for the loss of the dopamine that you would otherwise have there. And you can also use growth factors. You get these other proteins called growth factors, which we know stimulate cell growth. So you get ones that are brain-derived growth factor or glial-derived neurofactor, more commonly known as GDNF, which people may have heard of, because that's been around for a few years now, trying to develop that as a therapy for Parkinson's disease. And by putting GDNF in the brain, it supports or regrows sort of dopamine producing cells, and helping, the hope is to help that recovery, basically. And also there is the potential for modifying disease causing genes. So as I said, some people will have a genetic basis for their Parkinson's disease. So one such mutation is called GBA, or glucocerebrosidase. It's a difficult word to say this time of night. And that is a particular enzyme in the brain, and sort of that reducing that activity makes those cells less able, the dopamine producing cells less able to survive. So it may be possible to put in a new version of the GBA into the brain, and help reduce the impact of that mutation. So lots of different therapies being explored at the moment. And then in addition to gene therapies, both for Huntington's disease and Parkinson's, one of the other things that we can potentially do is replace those lost cells, so cell replacement therapy. So we saw the very specific regions, both in Huntington's and in Parkinson's, where cells are lost. So what happens if we take similar cells, put them back in that area, and the idea is that by putting cells back in that area, they will grow, they will do something what we call differentiate. So they'll, it's basically another word for maturing into the right kind of cell. And the hope is that those cells differentiate and form the lost connections and reform those circuits that we've been talking about. So this is a little bit more novel. So you can use what we call donated cells, so donated embryonic stem cells that are obtained from donated fetal tissue. So that has an ethical issue around it, so ethical consequences. But also the technology is now advancing where we can engineer what are called pluripotent stem cells. So we can engineer cell lines that are sort of like baby neurons, So they're not neurons yet, because if we try to transplant neurons are very delicate beings, and if you try to transplant a neuron, they would just die. So we take effectively what are baby neurons, but ones that we know are going to change into the neurons that we need. So for Huntington's disease, that would be those medium spiny interneurons. For Parkinson's disease, it'd be those dopaminergic neurons. And the hope is that we can put them in the brain and they will restore those circuits and sort of recover lost function. And I've just put a big question mark over this because it's still quite a new field. And there are only, I think there's one trial at the moment running between the UK and Sweden for a new stem cell therapy product in Parkinson's disease. There's a trial in the US, and there's an active trial in Japan as well. And there are cell therapies being developed for Huntington's disease as well, but they're yet to go into trial. So as I outlined earlier, there are some specific challenges for evaluating ATIMPs in neurodegenerative disease. I'm just going to run through a few of those now. So clinical trials, by their very nature, are long and difficult. So most people are familiar with the fact that it takes a really long time to develop new therapies, go through all the stages of clinical trials, and actually get a new therapy to market. But there are certain things, aspects of ATIMPs that make them a little bit more difficult. So one is delivering the therapy to the intended target location. So when we're talking about the striatum or the basal ganglia, they're really quite deep structures in the brain. So it's sort of way down in the middle, sort of towards the back. So pretty difficult to get to. And how do we actually get that therapy into the brain? So there's a lot of work at the moment going around focusing on how we effectively deliver those therapies into the brain. So very early on, when people were looking at this, it was essentially just a big needle, but you do get problems with what we call reflux. So if you inject something into the brain, because of the way that the brain is, and it's a bit of a jelly-like substance, when you take that needle back out, because you've got this nice track where you've put your needle in, when you take it out again, everything just rushes back up that track. So your therapy doesn't stay where it's supposed to be. So there are real issues around what device you use, and if you're considering using cells, we have to be really careful about how we inject those down a device. Cells are quite fragile, so you don't want them to sediment in a cannula or a needle. You don't want the shear forces to split those cells open, because you want live cells to go into the brain. And then there's something called the fidelity of delivery. And what we mean by fidelity is how well have we done it? So we can know that we've got the needle in the right place, but how do we know that what we're delivering into that area is actually there? And so with some of the gene therapies, you can do this. You can use MRI guided delivery. So you put a special, what we call a contrast agent, into that fluid that we're injecting. And that is, when we say contrast agents, that's something that shows up on MRI. So MRI is a way of taking pictures inside your head, in your skull, using a very strong magnet. And so when that's being infused in real time, you can take MRI images of the brain and see this fluid filling the space that it's supposed to. That's not possible for cells, though, because that contrasting agent would kill the cells. So we have to think about other ways that we may be able to do that. And these procedures take a really, really long time to do. We're talking, between 6, 12, maybe even 18 hours to perform a surgery for these. So these are the considerations that we really need to have for the safety of the patient, and then how we develop them as therapy so that they can be used within healthcare systems. The other thing, another thing is that these ATIMPs, they're going to require a really, really long time before we think that they're actually going to work, before we can notice that they've got a functional benefit. So if we put baby cells, these baby neurons into the brain, they need to take time to mature and grow into the areas that they're supposed to and form those connections. So we're really talking months and months and years rather than days and weeks to see any real difference in terms of function. So that's symptoms improving over the long term. Then when we're talking about doing cell therapies, there are real issues around where we get the cells. So understandably, donated fetal tissue is extremely scarce. And it means there are real problems in getting enough cells to be able to do a transplant. That's also one of the reasons for developing sort of pluripotent stem cells, in that we can manufacture those and grow them up under laboratory conditions. So we have to do it under what is called GMP, so good manufacturing practice. So it's all very highly regulated, and then they can be stored as and when they're needed. But there are real problems around the sort of manufacturing. That's a whole other research area in itself, how you scale up that manufacture. And then when I've got storage here, it's about how those are stored at the place where they're going to be used in a patient. So we really have to think about the ease of using them. So at the moment for ATIMP trials in neurodegenerative disease, they really only happen at a few specialist centres around the world. CAR-T, which is an approved and authorised therapy, again, still only happens in specialist centres because you need a lot of specialist equipment and expertise. Also, there are issues around participant selection for trials. How we have this very difficult playoff between including people who have disease that is advanced enough that should anything go wrong or there be a safety issue, they don't have, it sounds awful, as much to lose as someone who's earlier in their disease course. However, there is also the flip side of that, is that we may need to intervene really early in the disease course for these therapies to have the best effect. And it's likely that participant selection is going to change as these therapies develop and go through the phases of clinical trial and we establish safety first beyond anything else. And then for HD in particular, so HD is a rare disease. It means we've got a low number of participants to begin with. And how do we do lots of really robust studies when we have very few people to start with? And this is really important. ATIMPs are irreversible. If you put a cell or a gene product or a virus containing a gene into somebody's brain, you can't take it out again. That means that people may not be eligible for any kind of clinical trial in the future, and that could be really important to them. So it's really important when we're asking people to take part in these trials that we really give them the information that they need to make a truly informed decision. But I just want to spend just this last little bit saying, but what do the people who live with neurodegenerative disease think about these? So just talking about the participant experience, and because this is a particular area of interest of mine. So clinical trials, not only are they difficult to do, they're really difficult for people to take part in. And we should never forget how difficult it is for people to take part in them and the lengths that people go to in volunteering for these trials. They're extremely courageous. They give a lot of themselves, and that needs to be sympathetically addressed in our research. We've already discussed that we need to keep these people in these trials for a really long time, not only so we can measure how well that therapy may be working, but because these are so early in development, we really need to stay in touch with these people and monitor them for safety. And then we would argue that the trial participants, the people who've been through this process of receiving a cell or a gene therapy, they are the best advocates for these future therapies. If we understand their experience, we can support their ability to advocate to other people in the community, and they can provide early insights into the product as well as the trial, which is really, really important for product and intervention therapy development. And so understanding experiences from people who've taken part in these trials, it provides super, super important feedback around the trial design, how we put together a research program to evaluate these therapies, how the trial is conducted. So it's how we go through all sort of the logistics and the nuts and bolts, including that informed consent process. What people want to know, when do they want to know it, who do they want to hear it from, how do we get them to appointments, how many appointments are too many, do they want more appointments, are people willing to travel? If they're not, how do we make it easier for them so that they stay in the trial? Because one of the problems that the two major problems of clinical trials are always recruitment and retention. So how do we get people to take part, and how do we keep them taking part? Because it's really important people stay in a trial so that we don't lose what we call the power. So we need a specific number of people to take part and stay taking part, so we're able to answer our question really rigorously. So it's really important that we understand how participants are feeling about the process in order to try and keep them engaged and keep them in that trial. And also about what we measure. So when we talk about outcome measures, that's how do we measure if a therapy is working? Participants can tell us whether or not the tests that we're doing really reflect their experience of living with their condition, or their experience of how the treatment in the trial has affected their disease. So yeah, I would super advocate for really engaging with participants to understand trials. And that just about wraps me up, and I'll be really happy to take any questions. Thank you.
Many thanks indeed, Cheney, for that superb overarching review of these exciting new therapies. So I'd ask our participants to start sending in or continue to send in their questions. We've already got a few that have arrived, but please continue to do so. Cheney, if I could just maybe start off. Cardiff University has a close association, of course, with Archie Cochrane, who is a big proponent of the randomized controlled trial. So how can we justify ethically doing what essentially is surgery then to achieve the gold standard of an RCT for these kinds of therapies? I mean, how has it been done for some of the therapies that are already being used for Huntington's with the anti-nucleotide sense nucleotides, but how is it going to work for?
Yeah, that's a really great question. So what we're talking about here is what we call sham surgery. So a sham surgery is where you go through all the steps of having an operation without actually having the operation or receiving the treatment. So for cell and gene therapies, that would mean taking a person into an operating theatre for the length of time that you would expect them to be there if they were receiving the therapy. So that's, as I've said, 12, 16, 18 hours, having them under anaesthesia for all of that time, drilling what we call partial thickness burr holes into the skull. So you don't go right through, but you make enough of a dent in their skull so that if they were to put their hand on their head, they wouldn't know whether or not, so they would know that something had been drilled into their skull, but they won't know whether it's gone right through. It's a very, very ethically contentious issue. So there is actually even a difference in the regulators, the drug regulators, in whether sham surgery is acceptable or not for these types of trials. So in the USA, the FDA really are very keen on sham surgeries for gene therapy trials. In Europe, that's not quite the case. So the EMA is more reluctant to undergo sham surgeries. There are cultural differences as well, in that participants in the US seem to be perfectly happy with the concept. It's all right. But the feedback we've got from people with lived experience in the UK and in Europe is that that is absolutely not a goer, particularly as these therapies are at such an early stage. And at the moment, we're still establishing safety rather than whether or not they work. So there are other trial designs that we can use that don't include a placebo control. So the reason for including a placebo control is to reduce or minimize the amount of bias that we have. So if you know you're taking a pill, you're more likely to get better. And we know the placebo effect can be really high. And with placebos, the more risky the intervention, or the more sort of perceived risk around the intervention, the higher the placebo effect is. So we conducted a transplant trial, a fetal cell transplant trial in people with HD, and we used something called a trial within a cohort design. So we had a large group of people and we applied all the same tests to them, but then we just selected a few people out of that group without the other people knowing that they weren't selected for the intervention. And that's how we reduce that bias, basically. Right. So there are novel methods that we can use that don't include a sham surgery.
Thanks very much for that answer, Cheney. So if I start picking up some of our questions that have come in, does it require multiple doses out of chance or another reason? So I think that might have been referring to one of the early trials with the antisense.
Yeah, so I think that refers to the ASOs, the antisense oligonucleotide therapies. It requires multiple doses because your body is creating, so the cells in your body are creating those proteins all the time. They're constantly turning over. So if you want to stop that production, you have to keep putting that in. And as I said, ASOs, they're not hugely stable, so they're not going to hang around forever. Your body will break them down, and that's why you need to keep having those repeated doses. But if you've got it in a gene therapy in your head, that gene therapy also keeps being produced. So that's just a one-off administration if you have it direct, if you have that kind of technology put intracranially.
Okay, thank you again. I think you've touched on it already, but what stage are we in terms of actually treating the public with these therapies? I know you did mention there are a few trials already underway.
Yes, so the trials that are running at the moment, both in cell and gene therapies, there isn't anything past what we call phase 2. So you tend to have, there are 4 phases of clinical trials, but three that lead up to a drug being approved. So phase 1, very, very small, where we make sure that it's the new treatment is safe. Phase 2 is what we sort of refer to as proof of concept. So it's in more people, but not enough to absolutely prove that treatment works. And phase 3 is where we have these really, really big trials with lots and lots of participants that are very robust, very rigorous, and we can be absolutely sure then that any effect that we see, treatment effect, is a true effect of treatment. So most of the therapies we have are at phase 2 at the moment. Particularly with some of the cell therapy trials in HD that we think will be coming through in the near future. Our opinion is that we're going to need lots of small iterative trials rather than these big phase 3 trials, particularly with HD being a rare disease. So, you know, everyone talks about sort of 15 to 20 years. It's still going to be quite a long way off before they're actually therapies in the clinic. But something like HD has what we call orphan disease status with the FDA, which means it gets kind of an accelerated review because it's a rare disease, because it's fatal, because there's such a pressing need for anything that's disease modifying. There's an agreement that diseases that have this orphan status will get reviewed quicker.
Okay, thank you. Do these therapies aim to help or cure disease in their current status?
Yes, it's another really, really good question. So the aim, so as I said, for both Huntington's disease and Parkinson's, we don't have anything what we call disease modifying therapies. So when we say disease modifying, that's changing the disease in any way. So ultimately what we want is a cure, but we want something that will slow down the disease as well, and so slow down that progression. And the hope is that these therapies will slow down that disease progression to such an extent that you can live a normal lifespan. So effectively, it would be a cure, but it's going to be very difficult to effect a true cure in the purest sense. But certainly any kind of disease modification would be good as a starter.
I guess with something like Parkinson's, Cheney, there's obviously an ongoing disease process that is destroying the dopamine.
Yes, absolutely.
Neuron. So if you put in replacement cells, then presumably you're not going to be stopping those processes. But is the hope that treatments will be suffice for a number of years? Or
that raises another really interesting issue around sort of cell and gene therapies, is that we know what we call the microenvironment in these areas can be unfavorable for normal cell function and cell growth. So it may be, once we've got a sort of a proof of concept that we can get cell therapies to work and they do what we think they're going to do, is that that support that environment. So it may be that you want to put in cells with growth factors, or there are other issues about cell overgrowth that you don't want to generate. But it may be that you can add in other drugs that help support that cell growth and development. So
a couple of shorter questions. What are the costs per patient, and how do you test how successful a therapy might be?
So these therapies are so cell and gene therapies are going to be incredibly expensive. I had looked this up, and I always forget the exact figure. But the ASO therapy for SMA is in the millions of dollars for one person. There is currently a trial, a gene therapy trial running in HD at the moment, where the rough cost of that trial, and trials are more expensive than when a therapy comes out, but it's roughly running we're talking maybe millions per person rather than for the whole thing. They are going to be very, very expensive. However, there is nothing else that's disease modifying, and so there are going to need to, if these therapies are found to be effective, we're going to have to find some way of having those conversations about cost versus benefit, because of course if you've improved people's quality of life and the number of years that they can stay healthy, stay functional, then that has health economic benefits as well. So we would need to do sort of evaluations around that as well. And there was a second part to that question, wasn't there?
So how do you then assess whether the treatment has been successful or not?
So again, that's a really good question, and it's different between different diseases. Sometimes the drug regulator will tell, so we have to pick in clinical trials, we pick something called a primary outcome. And so that's one test that we do, and the success of the trial is based on whether or not we meet our primary outcome, whether we can show that we've that our therapy is going to have the effect on that measure that we think it will, or hope it will. So for something like Huntington's disease, it's often a measure around motor function, because it's easy to measure, and it's really obvious, and it's one of the main characteristics, sort of main symptoms of the disease. In a recent ASO trial, their primary outcome was a sort of took into account changes in motor function and cognitive function. So sometimes in Parkinson's disease, it's often what we call the UDPRS, which is essentially a rating scale for how bad someone's motor symptoms are. But these scales can be really subjective. So it depends on one person looking at another person, deciding where they are on that scale. That's subject to a lot of bias, and bias in clinical trials is bad. You want to avoid it at all costs. So there is a lot of work going on in developing objective measures, using things like what we call biomarkers. So that's where we can measure changes in someone's brain using imaging. People are trying to develop biomarkers looking at levels of various proteins in the cerebrospinal fluid. That's another way that we can look at success. And digital biomarkers as well. So using sort of remote digital technologies, so that we get sort of constant passive readouts of people's function rather than just testing them in isolated occasions.
Thank you again, Cheney. Cheney, we're going to have to be a little selective with the questions. I know time is running out. What is the role of the Centre for Trials Research? Do you arrange for patients to join trials in the UK and elsewhere? And how do you recruit your patients?
Yep, great. So Centre for Trials Research, we are an academic clinical trials unit. So we design we have lots of different divisions. So I'm in the brain health and mental wellbeing division. So we look at neurodegenerative diseases, mental health, healthy aging. We have a webpage on the Cardiff University pages that lists all our active trials. We usually recruit through either GP surgeries or hospitals. That's where we go out and look for our specific populations. So lots of different ways of getting involved in research. I would say if you're interested in getting involved in research, is to either talk to your doctor. If you've got a chronic condition and you have a specialist, they should know any active trials that you can get involved with in your region. And also if you're interested in sort of being involved in research as a general member of the public, or giving of your experience, then contact Healthcare Research Wales Involve. And that's a really good way of getting into being involved in research.
Great, thank you again. Cheney, I'm going to finish up with one more question here. And does your remit for research in the neurodegenerative diseases translate to many other similar health conditions? Is there any scope that you may consider expanding the remit to include conditions like ataxia?
Yes, absolutely. So there are, so if I walk back right to sort of the beginning when I was talking about Huntington's disease, and that sort of the mechanism by which the disease happens is that CAG repeat. We call it a CAG expansion. There are other genes that have that same problem. So the spinocerebellar ataxias are triplet repeat disorders. And there is research actually in the university going on into how we can control the expansion of those CAG repeats, those unstable repeats, either with medicines or with gene therapy. And that technology would be applicable to other neurodegenerative disorders as well. And there are other neurodegenerative and neurological disorders where gene and cell therapies are being developed. So in epilepsy, they're being developed to help people with treatment-resistant epilepsy. I could go, there wasn't enough time to include all of them, but there are also cell therapies that you can use in multiple sclerosis as well. So there's a really wide remit.
Yeah. So I'm really aware that some excellent questions that we haven't been able to get to this evening, Cheney, but I see you've given us your email address. Would it be okay for some of our participants to email you directly with their questions?
Absolutely.
A number of specifically asking whether there are any specific papers or suggestions where they can look for more information, that kind of thing. So if they contact you directly with the email address, that will be when we post your talk online, your email address will be in the final slide, and then people can contact you directly. Okay, well, thank you once again, Cheney, for a really, really stimulating talk this week, this evening. I'm sure there'll be some taking encouragement from where this field is going, and it'll be very interesting to see how things do progress in the coming years. So, very many thanks again for your presentation this evening. So, just to remind our participants, you will be receiving a text shortly requesting feedback on this evening's presentation. So please look out for that. And if you'd be kind enough to give us your feedback, that would be very valuable. We have our next talk coming up in January. So we're back again in early January when we have Professor Dan Davis from Imperial College. I don't know if we have a slide for that, Karen. Highlighting next month's talk, but hopefully many of you will be back with us in early January for that next talk in our series. So it just remains for me to say Nadolig Llawen, Merry Christmas to everyone. Thank you for your excellent questions and engagement this evening, and we look forward to seeing many of you again with the 3 remaining talks that we have in the series. So Diolch yn fawr iawn. Thank you very much again, Cheney. Very much appreciated and valued this evening. Okay. Good night, everyone. Bye-bye.